Okay, guys, I hope you are fine today. So today we are looking at physiology of blood, lecture 4. So we are still dealing with physiology of blood units. So today we'll be looking at lecture 4. So in this lecture, what are we going to look at today? Okay, so I hope you are getting me. So in this class, we're going to look at blood typing. Then we'll move on to cross-matching and transfusion. Then we'll also look at rhesus factor and incompatibility. So rhesus factor and incompatibility. So these are the things that we'll be discussing in this lecture. So it means by the end of this class, the objectives should be able to describe blood typing, different types of blood typing, the major ones that we'll be discussing, and also you should be able to describe cross-matching and transfusion. And you should also be able to describe rhesus factor and incompatibility. So generally, this is what we'll be looking at today. So I hope you are fine and you are able to get my class as we proceed. So just relax and just remain focused and enjoy. So we'll start by looking at blood typing, cross-matching, and transfusion. So these things are related. So that's why I'm putting them here. But before we proceed, let's look at the basis of blood grouping or typing, the multiplicity of antigens in blood cells. So this multiplicity of antigens in blood cells, I remember when we are looking at blood physiology or cell physiology in general, we say that cells, they have plasma membrane, and that plasma membrane is composed of proteins, lipids, and carbohydrates. Most of it are proteins and lipids, then less of the carbohydrate. So the lipids that are found within the plasma membrane are called phospholipids, and they're the ones that were forming the phospholipid bilayer, separating the intracellular from extracellular fluid. But you also realize to say that we're also looking at plasma proteins or plasma membrane proteins. These are proteins that are associated with the plasma membrane. So these proteins, some of them were functioning as organ of recognition. We said an example that we gave was an antigen. So antigen are some of the proteins that are found within the plasma membrane, and they will facilitate the immune cell to be able to recognize self. So how do the immune cell know to say that this is part of me, this is a foreign object, and then they will mount an immunity against it? It's by looking at these antigens that are found within the plasma membrane of different cells. But since we're looking at blood typing, there are specific antigens that we'll be discussing today. So the ABO system was discovered in 1901 by Landsteiner. So this is a scientist that we are looking at antigens that are found within red blood cells. So he's the one who described the ABO system, which is very important even now. So this is the scientist that described the ABO system. He is called Carl Landsteiner. So this is a scientist that described ABO system. At least 30 commonly occurring antigens, which are called agglutinogens, have been found on the cell membrane of red blood cells that can react with plasma antibodies or agglutinins. So on these red blood cells, there are over 30 antigens that have been identified. And these antigens, they are called agglutinogens. So the antigen, it ends with GEN, and agglutinogens, they also end in GEN. So how do you know to say that antigens are also called agglutinogens? It's just by looking at the way this word ends. So you can see antigens, they end with GEN. Then the antibodies, which are different, they always end in NIN. So these are called agglutinins. The antibodies are also called agglutinins. The antigens, they are also called agglutinogens. So there's a reaction between the antibodies and the antigens, or a reaction between agglutinogens and agglutinins. So the antigens are proteins that are found within the plasma membrane of red blood cells. There are over 30, that have been identified, but the common ones that we'll be discussing here are ABO antigens and the rhesus antigens. Those are the ones that can cause transfusion reactions. So according to the presence and absence of these antigens, blood is classified into several groups or types. So blood typing, blood grouping, you're looking at the type of the antigen that is found within the plasma membrane of red blood cell. So the plasma membrane of red blood cells, they'll have different antigens. So depending on the presence and the absence of these antigens, we are able to classify blood groups. So you have different types of blood groups depending on the absence and the presence of these antigens. So those that will have antigen A, then is classified as blood group A. Those that will have antigen B, they are classified as antigen B. Those that do not have antigen A or antigen B, then they are called blood group O. So they don't have these antigens, but it's not really correct to say that the blood group O, they don't have antigens on their plasma membrane. That's not correct, because even blood group O, they have an antigen, but it's just that the antigen that they have do not stimulate the immune response. So you have a special type of antigen, which is called antigen H or H antigen, that it will form the backbone for other antigens, as we'll be discussing later on. But you know to say that this H antigen do not stimulate the immune response against it. Because of that, sometimes we just say that blood group O, they don't have antigens on their plasma membrane, which is not correct. The two major groups of antigens that can cause transfusion reaction more than the others are the ABO antigens and the rhesus or D antigens. So these are the antigens that can cause transfusion reactions. So they're the ones that we'll be discussing in this lecture. So let's look at the genetics of ABO antigens. So like I said, there's the H antigen. So this H antigen is a structural backbone of A and B antigens and is present in almost all people. So each and every person, they express this H antigen, but this H antigen is recessive. So in presence of the other antigens, then you'll find that the phenotype will be expressed depending on these other antigens apart from the H antigen. But what you need to know here is that the H antigen will form the backbone for A and B antigens. So in terms of structure, you find that the A and B antigens, they will have a backbone of H antigen. And then on top of that, depending on the type of a carbohydrate molecule that is added to it, it can either be A antigen or B antigen. So we'll look at the carbohydrate moieties or molecules that can be added to the H antigen for it to be transformed into A or B antigen, depending on the carbohydrate molecule that is added. So you have two carbohydrate molecules that can be added to H antigen. So you have galactose for B antigen, and then you have N-acetyl galactosamine for A antigen. So we'll describe all that as we proceed with our class. The O gene does not produce an active transferase, and so the H backbone remains unaltered or unchanged and is not antigenic, meaning that the gene that codes for H antigen, it doesn't produce any enzyme, a transferase enzyme that is capable of transferring a carbohydrate moiety from a donor molecule to a recipient molecule. The donor molecule is the one that will contain that carbohydrate moiety, and the recipient molecule in this case is the H antigen. So you find that people who are blood group O, in their gene, there is no information for coding for this transferase enzyme. Because of that, they are unable to transfer these carbohydrate moieties to H antigen. So for this reason, you'll find that there's only anti-A and anti-B, but they will never be anti-O. the antibodies that will be produced, it's either you can have antibody A or antibody B. There will never be a point whereby somebody will produce anti-O. Why? It's because we said the H antigen does not stimulate the immune cells to produce antibodies against it. So you will never find O antibodies or anti-O. The A and B antigens are carbohydrate dominants of glycoproteins and glycolipids, distinguished by the type of sugar molecule added to the backbone of the antigen, the immunodominant sugar. So these are the sugars I'm talking about. So these sugars, they are also called immunodominant sugar. Why? It's because they are going to stimulate the immune system, the immune cells, to start producing antibodies against them. So depending on these carbohydrate sugars, then you have different types of antigen. On top of the H antigen, that will form the backbone of all the antigens. So if you have N-acetylgalactosamine, this will code for group A. Galactose is for group B. So if you add N-acetylgalactosamine to H antigen, then to become anti-A. If you add galactose to H antigen, then you have anti-B. So those are antigens. So the antigen for A, on top of the H antigen backbone, it will also have N-acetylgalactosamine that has been added by this enzyme, which is called a transferase. Then if you add galactose, then this H antigen will be transformed into B antigen. The A and B genes found on chromosome 9 encode for glycosyltransferase. These are the enzymes. The glycosyltransferase are the enzymes that are responsible to transfer these carbohydrate molecules or moieties to H antigen. So the glycosyltransferase, they can transfer N-acetylgalactosamine to H antigen to convert it into antigen A. Or it can transfer a galactose to H antigen to transfer it into antigen B. The glycosyltransferase categorize the transfer of appropriate immunodominant sugar from a nucleotide donor to an acceptor substrate. And this acceptor substrate is the H antigen because it's the backbone for the other antigens that we are discussing. So this is what I'm talking about. So you can appreciate in this diagram here, you have red blood cell. This red blood cell has a plasma membrane or the cell membrane. On the cell membrane, you have these glycoproteins and glycolipids. And depending on the carbohydrate moiety that is attaching to those glycoprotein and glycolipid, in general, you have the H antigen. So this is the structure of the H antigen. This H antigen is composed of these glycoprotein or glycolipids, but the carbohydrate part that is attaching to a protein or to a lipid, this is what you're interested in, that will form the antigen. So if you have glucose attaching to galactose, attaching to N-acetylglucosamine, attaching to galactose, and fucose, this is a structure of H antigen. This H antigen will form the backbone for other antigens. So what do I mean? Depending on a type of sugar moiety that is added to a galactose here, then it can be transformed into antigen A or antigen B. So if we add another galactose here to this galactose, then you're going to have antigen B. But if you're going to add N-acetylgalactosamine to this galactose here, then it will form antigen A. So you can see the H antigen, which is the backbone here, which is called O antigen or H antigen. So depending on what you are adding here. So that enzyme, a transferase, is the one that is going to transfer those carbohydrate group to this galactose here. So if it's going to transfer N-acetylgalactosamine to galactose, then this antigen will become A antigen. If it's going to transfer galactose to this galactose here, then it will become B antigen. So these are the types of antigens that are found on plasma membrane of red blood cell. You can have O antigen that does not stimulate an immune response. So in blood group O, it means that there will be no antibody against it. Then those with antigen A, this can stimulate an immune response. So there are antibodies that can recognize this group here. So those antibodies will now fight against the antigen A. So those antibodies against antigen A, they are called A antibodies. So the A antibodies or anti-A, they are antibodies against the A antigen. Then you also have antibodies against B antigen. They are called anti-B or B antibodies. So they are antibodies against the B antigen. This is the same diagram just expressed in a different form. So you can see here the H antigen for blood group O. This is the backbone for other antigens. So depending on what you are adding, if you add a galactose, it becomes, if you add N-acetylgalactosamine, then it becomes A antigen. If you add galactose, then it becomes B antigen, like I've already explained here. Now let's look at inheritance of ABO blood group. So the ABO blood group is inherited from both parents. So the male and the female, when they are mating, they will transfer information or the gene to the developing embryo or the zygote that is being formed here after fertilization. So depending on the combination of the genes that you inherit from the mother and the father, you have different types of blood groups that you can inherit from them. So what you need to know is that the gene that codes for A antigen and the gene that codes for B antigen, they are dominant genes. But the gene that codes for H antigen or the O antigen is recessive, meaning that whenever you have a combination whereby you are inheriting A antigen from your father and then you inherit an O antigen from your mother, it means that the phenotype of that blood will be blood group A. Why? It's because the O antigen or the H antigen is recessive. So the one which is dominant is the one that will be expressed, phenotypically. The other thing that you need to know is that these agglutinogens will appear during the 6th month of fetal life. So in the fetus, you won't appreciate these antigens on the plasma membrane of the red blood cells as it's developing in embryo form. But at 6th month of fetal life, then they will start expressing these antigens, meaning that a baby is born with red blood cell antigens on the plasma membrane because they start showing at 6th month. The concentration at birth is about 1/5 of the adult concentration. And it rises to adult level at puberty. So as the child is growing, you find that at puberty level, the concentration of these antigens will be almost of the same concentration to that of an adult person. Meaning that when the embryo is developing, they do have the gene that can code for these antigens, but those genes, they haven't yet transcribed into messenger for them to start producing proteins. So as an embryo, they only have the gene within their genome, but they are not capable of producing these antigens yet. So at 60th month of fetal life, that's when they will start appearing. At birth, the concentration is just 1/5 of that of an adult. Then at puberty, they have almost the same concentration of this antigen compared to that of an adult person. These agglutinogens are present not only in red blood cells, but also in many organs, like the salivary glands, the pancreas, the kidneys, the liver, and the lungs. So it's not only red blood cells that can express these antigens. Even other body cells, they can express these antigens. That's why you find that when there's an organ transplant, the immune system can reject that depending on these antibodies or the antigens that are found on the plasma membrane of different cells. That's why you get such reaction. These ABO carbohydrate agglutinogens are also present in cell membrane of saprophytic bacteria and pollen. So they are not just found on red blood cells, they are also found in bacteria and pollen. The ABO antibodies or agglutinins development, how are they going to develop? Because we said as the embryo is developing, they don't have the antigens. They will only have the antigens at 60th month of fetal development. Then later on, after birth, they will start developing them until puberty, when they will have the normal concentration of these antigens. So now, if the fetus doesn't have those antigens, how do they start producing antibodies against the antigens? So like I said, the agglutinins are IgM. So they are big, they are IgM. There are only 2 agglutinins. Okay, so we proceed. So the antibodies, agglutinins development, there is only anti-A and anti-B that are produced during fetal life. So that are not produced during fetal life. So these antibodies, they are not produced during fetal life. But when do they start being produced within the body? After birth. So most infants, they rapidly develop antibodies against the antigens not present in their own cells. So the neonates or the babies, once there is anything that is foreign, a protein that is foreign, you find that they'll start now producing immunity against those foreign proteins. So how does that happen? Because we say that in these neonates, you find that the development of antigens, they will start at 60th month of fetal life and then later on after birth. So how do now the antibodies come in? How do they start being produced? So since ABO carbohydrate antigens are also present on the cell membrane of saprophytic bacteria, which appears in the gut in the neonatal period and on the cell wall of the pollen grains. So because these ABO antigens, they are also present on bacteria, like saprophytic bacteria and also pollen. After a baby is born, you find that they'll start gaining these bacteria. So bacteria will start getting into the baby via the GIT, the gastrointestinal system, via the mouth, and also via the nose. You have introduction of these bacteria and pollen. And these bacteria and pollen on their plasma membrane, they have ABO antigens. So because of that, they are going to stimulate the immunity of the neonate to start producing agglutinins or antibodies against them. And that will start appearing only 2 to 3 months after birth, meaning that the production of antibodies will only start 2 to 3 months after birth. This will be stimulated because of the presence of saprophytic bacteria in the pollen that will also have the ABO antigens on the plasma membrane of these cells. So once they get into the neonate body, you find that the neonate will now start producing antibodies against them because they can't recognize them. They're not part of their cells, so they need to defend themselves. So they'll start producing. So now the developing immune system or the neonates will treat these agglutinogens as non-self, which are absent on their red blood cells, and they'll start developing the antibodies against them, like I said. So these antibodies are against the bacteria in the pollen grain antigens. So these bacteria in grain antigens are natural occurring antigens. So they are developing antibodies against the natural occurring antigens, and also called the natural occurring antibodies. So these antibodies, the initial antibodies against the natural occurring antigens, they are also referred to as the natural occurring antibodies. That's why ABO antibody systems, or the antibodies, they are referred to as natural antibodies within the body. Why? It's because they are produced against the natural antigens that are found within the bacteria and the pollen. So these antibodies equally react with human antigens. So in as much as these antibodies that are being produced are against the antigen that is found within the bacteria and the pollen, but it can also react with a human antigen. So it is a good example of cross-reacting antigens. Cross-reacting antigens, the cross-reacting antigens are antigens that are meant to be produced against one organism, but it can react with another organism. In this case, the ABO antigens that are produced here, they are against the bacteria and the pollen, but you'll find that it can also react with a human antigens. So naturally now, they can also react with the ABO antigen that are found within the red blood cell of another person. So this is how the antibodies develop. This diagram here is just summarizing the blood group and the antibodies that are found in an individual. So looking at this table, you have blood groups on top here. Then coming this down here, you have blood type. Then you have antibodies in plasma and the antigens that are found within the red blood cells. So these blood groups, there are different blood groups. There's blood group A, B, AB, and O. These blood groups are grouped depending on the presence of the antigens that are found within the plasma membrane. So when you're looking at a red blood cell with antigen A on the plasma membrane, so the antigen that is found there is antigen A. It's referred to as blood group A. Why? It's because it has got antigen A on the plasma membrane. So if an individual is blood group A, what sort of antibodies is this person going to have in plasma? The antibodies in plasma, a person with blood group A will have anti-B antibodies. So you want these antibodies to react with blood group B or blood group AB. You don't want these antibodies to attack your own cells or to attack antigen A. So they are not against antigen A. They will be against antigen B that is found in blood group B and blood group AB, but it's not found in blood group O. So it can't attack blood group O. Then a person with blood group B will express antigen B on the plasma membrane. So the form of antigen that is found is antigen B. The antibodies that we find within plasma is anti-A. So that if you give blood group A to a person with blood group B, this anti-A or the antibody A will react with the A antigen because it's not found within the plasma membrane of the cells of that person. Blood group AB, you have both antigens. So you have antigen A and antigen B. So you have antigen B and antigen A. So because this person have both antigens, the blood group is referred to as blood group AB. This person won't have any antibodies within plasma. Why? It's because you don't want to have antibody B because it can attack the antigen B. You don't want antibody A because it can attack antigen A. So the person with blood group AB do not have antibodies in plasma. But later on, we'll also look at the rhesus factor. It can have an antigen or an antibody D, which is called the rhesus antigen that we'll discuss later on. But here we're just looking at AB or blood system. A person with blood group O, it means that they have H antigen or the O antigen. And the O antigen is not immunogenic. So if it's not immunogenic, it means it cannot stimulate immune system to develop an antibody against it. So there is no antibody against the H antigen or the O antigen. Because of that, sometimes we say blood group O, they don't have any antigen because this is the backbone for the other antigens, like we have already explained. When you're looking into the plasma of this person with blood group O, they're going to have both antigen, antibody A and antibody B, or anti-A and anti-B, meaning that if you give blood group A to a person with blood group O, these anti-A will attack the blood group A, or the red blood cell with the A antigen on their plasma membrane. If you give them blood group B, the anti-B will attack that blood. If you give them AB, both anti-A and anti-B will attack that blood. So you don't want that kind of a reaction. So this table in general is just summarizing the blood groups, the antigens that are found on the red blood cells, and the antibodies that you are going to find within the plasma membrane, and also the antigens. So it doesn't have an antigen. The blood group O doesn't have an antigen. But this is not correct because they do have an antigen which is called H antigen, which is common to all the cells, but depending on the sugar moiety that is added, then you have different types of antigen coming in, either A or B, depending what you're adding to the backbone, which is the H antigen. But in general, we say blood group O do not have antigen. This diagram is basically the same as the previous one, but it's looking at the genotype and the blood types. So you have the blood type here and the genotype. The phenotype is a blood group that that person will have. The genotype is the combination of the gene that are being inherited from the mother and the father. So let's start. The blood groups, blood type A, the combination of the genotype that you can find with a person with blood group A, it can have AA genotype or AO genotype. So like I said, AA genotype, AO genotype, in terms of blood group, there will be blood group A. Why? It's because the A antigen is dominant, the O antigen is recessive. So those that will have AA or AO, they will still be blood type A. Because here the O won't be expressed, you have more of the A because this is dominant. A person with blood group B, the genotype will be BB or BO. That of AB type, they will have the genotype. They will also have two genes, one coding for A, the other one coding for B. So the genotype is AB. Because A is dominant, B is also dominant, they will both be expressed. So you're going to have blood type AB. Type O, for a person to have type O, it means they need to inherit both genes from father and mother, which are O. So O gene from mother, O gene from the father, so that they are both recessive. And then the end result, you have the H antigen. this H antigen is not considered as an antigen because it doesn't trigger immune response. So it's as good as having no agglutinogen. So how do you inherit this? For instance, let me just give you an example. Okay, so let's say you have mother here and you have your father here. Then your mother, the genotype for your mother, it's A. That of the father, let's say it's B, O. So how are you going to inherit? So these two combinations, the mother and the father, when they have a child, the possibility of blood groups that their child will have or the offspring is a combination of A and B. That will be A, B, or you have a combination of A and B again here. So it will be A and B, or you can have A and O. So you have A, O, or you can have A, O again. So A, O. So you can see the blood type of, or the genotype of the offspring. It can be AB or AB or AO, AO. So 50% will be AB. So you have 50%. That will be AB, and also another 50% will be blood group A. So 50% A, and another 50% AB. So when you're looking at the genotypes, you have AB, AB, AO, AO. So this AO, AO, in terms of phenotype, it will be blood group A. Why? Because the O is recessive. So this is how you inherit that type from the parents, depending on the genotype of the parents. Okay, so we proceed. So let me just remove this and then we proceed. Okay. We proceed. Let's look at the relative frequencies of the different blood types. So in a population, you'll find that 47% will be blood group O, 41% blood group A, 9% blood group B, and 3% AB. So from these statistics, it is obvious from these percentages that the O and A antigen are frequently, whereas the B gene is infrequent, meaning that most people, they will have blood group O or A. B and AB, it's rare because of the percentages, meaning that in terms of in existence, the gene for B and the gene for The gene for B is rare, so it means that blood group B will be rare, and also blood group AB will be rare, because in AB you need a gene for B. So just not to say that most people, they'll be blood group O, 47%, and those with blood group A, 41%. And then blood group A, blood group B, it's rare, it's about 9%, and the AB combination is about 3%. So this question comes in MCQs or one-based answer questions, whereby I give you the percentages and then you are able to tell. So you need to remember this, that blood group O, they have the majority, 47%, blood group A, 41%, and then blood group B, 9%, AB is 3%. It's very easy to remember that. So in practical now, let's look at blood typing and cross-matching. So I'll speed up. Blood transfusion. Hemolytic transfusion reaction. So when you mix incompatible blood, you expect that there will be agglutination. Why? Because the antigen will start reacting with the antibodies. So the hemolytic transfusion reaction, the immune hemolytic transfusion reaction, is due to the incompatible blood transfusion. So due to incompatible blood transfusion, so if the blood is incompatible, you'll find that there will be a reaction because the antigen will start reacting with the antibody. This will result into destruction of donors' cells. So due to the specific arrow antibodies in patients' plasma. So in the patient, you have those antibodies against the antigen. So if you give the wrong blood type, you find that those antibodies will start attacking the antigens, and then that will result into an immune response that will result into destruction of the red blood cells. And then you have hemolysis taking place. So ABO incompatibility cases are more severe than the rhesus factor incompatibility. So because the ABO is more severe, so you don't want to miss out on this one when it comes to typing. So you don't want to give the wrong blood to a patient because there will be a severe reaction, hemolytic reaction that will take place. And later on, you also need to look at the rhesus incompatibility as well. So this can result into hemolytic anemia. Why? It's because when you give wrong blood to a person, you find that the antibodies will react against the antigens, and then they will attach to that red blood cell. So as the red blood cell is moving within the capillaries of the spleen, it will be very difficult for it to pass through that because of the antibodies attached to it, and then it will rupture, and then it will be phagocytosed by the reticuloendothelial system that is found within the spleen. And then you end up with a hemolytic anemia. Blood typing and cross-matching. So we've already looked at blood typing. So typing involves testing blood with non-antisera that contains antibodies, anti-A, anti-B, and anti-D or anti-rhesus factor. So when you're looking at blood typing, you need to type the blood against the antisera. The antisera is the one that contains antibodies. So for you to know to say this is blood group A, for instance, you get blood sample from a patient, you don't know what group it is, then you put it on a slide, then you add anti-A, antibodies. If there's a reaction, it means that these antibodies that are specific for A antigen, they are reacting with that. So it means that that blood group will be blood group A. If you put anti-B to that blood, you find that there will be no reaction because this antibody is not finding any antigen where it can attach to. Remember, these antibodies are specific for the antigens. So anti-A is specific for A antigen, anti-B is specific for B antigen, and rhesus factor is specific for D antigen. That we'll appreciate later on. So these antigens, they are found within the plasma membrane of the cells. So depending on the types of the antigens that are found within the plasma of the cells, you're able to type blood. So if you mix this blood with anti-A and then there's a reaction, then you will know to say that these antibodies that are specific for A antigen are reacting. Hence, the blood group for this person will be blood group A. That's how you type blood. Cross-matching is mixing of donor cells with recipient serum. So you're mixing donor cells with the recipient's serum. So the donor, you provide the cells, and the recipient, you provide the serum. Within the serum, you have antibodies. So this cross-matching It's a test that is done before transfusion. You want to see if the patient's blood will be able to react with the donor's blood. So that's why you are mixing the donor's cells, the red blood cells, with the patient or the recipient's serum. You remove the cells from the patient. Why? It's because if you mix donor cells with the whole blood of the patient, if there's a reaction, you won't be able to tell which cells are being attacked here, whether are the cells for the donor or the cells of the recipient. So by so doing, you won't be able to tell the reaction. So the only way you can tell the reaction is by getting the cells from the donor, just the cells, and the serum from the patient or the recipient. So that if you see that there is a agglutination reaction, you know to say that it's the antibodies from this patient that is attacking the donor's blood. So in so doing, you'll be able to say that this blood is not compatible. So you can't mix them. You can't do a transfusion for that. So mixing of incompatible blood causes agglutination, a visible clumping of blood. So you find that those antibodies will start attaching to the antigen. So because these antigens, they have more than one binding site, so they can bind to more than 2 red blood cells. Because of that, you can see that you are able to see blood clumping here or forming agglutination. Why? It's because the antibodies, they have more than 2 binding sites, so they can bind to this red blood cell and another red blood cell. That will result into clumping of blood, which is called agglutination. Under microscope, we can appreciate the cells that are agglutinating because of those antibodies that are touching different types of red blood cells. So this one is also different from Rolex formation. Rolex formation, you find that when you just add normal saline, it will resolve itself. But when there's agglutination, even if you add normal saline, these cells won't separate because they are attached by antibodies. Okay, so this is how blood cross-matching is done. So you have the recipient's blood or the patient's blood where you have the serum, and then you have the donor's blood where you get the red blood cells with different blood types. So for instance, if the recipient blood is type A serum, meaning that this person has got type group A or blood group A, then the serum will have antibody B, like we said. Those with A blood group will have anti-B in their plasma membrane. So since you're using the serum that contain antibodies, you're only going to appreciate anti-B for this patient. So if you mix this serum with type A blood, the type A blood, they have antigen A. So because they have antigen A, you won't get a reaction here. So it means that these blood are compatible. So you have type A, which are compatible. So they can be mixed or transfused. But if you get the recipient's blood serum with type B, they'll have anti-A, then you mix it with type A, meaning that the red blood cells, they have A antigen, and then in this blood you have anti-A, so there will be a reaction here, there will be agglutination. So meaning that blood type A and type B, they are not compatible here, as you can see. So it's the same as you appreciate from the other examples down here. So you can see here, there's a reaction. If there's a reaction here, it means that the red blood cells for this donor's blood, you have the antigen B, which is reacting with this antibody B. And here, there's no reaction. Why? It's because you have antigen B, here you have anti-A, so there will be no reaction. It's compatible here, blood group B and blood group B. The other one here, AB, you'll find that here there'll be a reaction. Why? It's because AB, they have both antigens, antigen A and antigen B. So if you mix with this serum that contains anti-B, the antibody B will react with the antigen B of the AB blood type. So there will be agglutination. If you mix it here again, you'll find that they also have antigen A. So the antigen A from this serum will react with the antibody A will react with the antigen A, and then there'll be also agglutination. This is cross-matching, which is different from blood typing. So blood matching or blood typing is a laboratory test done to determine the blood type of a person, like I've already explained. When a person needs blood transfusion, another test called cross-matching is done after the blood is typed. It is done to find out whether the person's body will accept the donor's blood or not. So after doing blood typing, you know that the blood are compatible. You also need to do a cross-matching because sometimes there are also other antigens that that person can react to apart from the ABO antigens. So you need to do a cross-matching to make sure that these blood you can transfuse. Otherwise, if you are getting a reaction after cross-matching, then you are not supposed to do a transfusion. So for blood matching, red blood cells of the individual or from the recipient and test sera are used. So for you to type blood, what you need is the test sera and the red blood cells of an individual. So depending on the test sera, these are antibodies. You have anti-A and anti-B. Then by looking at the reaction, you are able to type the blood. If it's reacting with anti-A, anti-sera, then that would be blood group A. If it's reacting with anti-B, anti-sera, that would be blood group B. If it's reacting with both anti-A and anti-B, anti-sera, then that would be blood group AB. If there is no reaction, that would be blood group O. So that's what you do when you're talking about blood matching or blood typing. Cross-matching, like I've already explained, is done by mixing the serum or the recipient and the red blood cell, the donor. So the recipient, you get the serum. Within the serum, this is where you find antibodies. Then you mix with the red blood cells of the donor and you see if this serum will react with the red blood cells to ascertain whether it's compatible or not. Cross-matching is always done before blood transfusion. If agglutination of red blood cell from a donor occurs during cross-matching, the blood from that person is not used for transfusion. Matching, you are using recipient's red blood cells and the test sera. Like I said, for cross-matching, you're using the serum from the patient and the donor's red blood cell. Okay, so this diagram, I think, is the same like the previous one I was explaining to you. So you're just checking for compatibility of blood. So you have blood type here and the serum. So the blood type, you can have blood group O, A, B, or AB, depending on the antibodies or the antigens that are found in the plasma membrane, the antigens that are found in the plasma membrane. So if you have antigen A, then the blood group is A. If you have antigen B, the blood group is B. If you have both antigen A and B, the blood group will be AB. If you only have the H antigen, it's as good as not having any antigen, and then it will be O blood group. The serum will have different antibodies depending on the blood type. So if you're looking at blood type O, they'll have anti-A and anti-B antibodies or agglutinins. Then if you have blood group A, you have anti-B antibodies or agglutinins. If you have blood group B, you have anti-A antibodies, AB, there are no antibodies. So when you're mixing this blood, if you mix blood group O to a person with blood group O, there will be no agglutination. Why? It's because this red blood cell doesn't have antigens on the plasma membrane. So these antibodies won't attach to anything. So there will be no agglutination. So if you give blood group A to this person with blood group O, what will happen is that this person with blood group O, they have anti-A and anti-B. So the anti-A will attack the antigen A from the blood group A. So there will be agglutination. So they are not compatible. If you give blood group B, you have B antigen. Here you also have the anti-B or antibody B that will react with the B antigen. So there will be agglutination. If you give AB, both anti-A and anti-B will react with antigen A and antigen B respectively. So there will be agglutination. Then when you have blood group A, you have antibody B. This antibody B, if you give blood group O, it means there'll be no agglutination because this antibody B is specific for B antigen, and you won't find it here. So there'll be no agglutination. If you give blood group A here, the blood group A that has got A antigen, this antibody is against antigen B, so there'll be no agglutination because there's no B antigen to which this antibody can react to. Then if you give blood group B here, what will happen is that you have the B antigen that will react with the B, you have the B antibody that will react with the B antigen, so you have agglutination. They are not compatible. If you give AB, the antibody B will react with the antigen B because here you have both antigens, A and B, so there will be agglutination. They are not compatible. Come down here, blood group B, they are going to have anti-A antibodies. If you give them blood group O, there are no red blood cells, there will be no reaction here, no agglutination. If you give them blood group A, this person has got antibody A against the antigen A. So you expect that there will be agglutination here, reaction. Why? It's because this anti-A antibody will react with antigen A that is found on the plasma membrane of this blood group A. So there will be agglutination. If you give blood group B or red blood cell B, they have B antigen. Here you don't have the antibody B. There will be no reaction, so they are compatible. If you give AB, you have antibody A. In AB blood, they have both antigens, so there will be agglutination because the antibody A will react with the antigen B here, so there will be a reaction. Then if you have AB blood group, they don't have antibodies to react with the antigens. So if you have AB, you give them blood group O, there are no antibodies, there will be no reaction, no agglutination. If you give them blood group A, there are no antibodies to react with the antigens, no agglutination. You give them blood group B, there are no antibodies, no reaction. If you give them AB, the AB blood, they have antigen A and B. But in this person plasma, there are no antibodies to react with any antigen. There is no agglutination. That's why we say that blood group AB is a universal donor, then blood group O is a universal, blood group O is a universal donor, and then blood group AB is a universal recipient. But you need to pay attention because there is another antigen that is not explained here, which is called the D antigen or the rhesus factor that will change. And because of that, you find that blood group O alone is not just the universal donor, because some O blood group, they can have antigen D or the rhesus factor that can also react. We'll explain that later on. Okay, so this is cross-matching, the way it's done in the lab. So you have the recipient serum that is mixed with donor's red blood cells. So you get the serum where you have antibodies, and then you get the red blood cells. You mix 2 drops of the serum with 1 drop of red blood cells from the donor. Then you do the washing here. So if these antibodies are specific for the antigen that is found within these red blood cells, they are going to attach. But if they are not specific for this antigen, there will be no antibody-antigen complex that will be formed. Then you do the washing 3 times, and then you incubate at 35 degrees Celsius for 1 hour. So why do you do the washing? You want to wash out the antibodies if there is no reaction. So if there is no reaction between antibodies and antigens because they are not specific, then the antibodies will be washed out. You will just remain with red blood cells. But if these antigens are specific for these, these antibodies are specific for these antigens, when you mix them, they're going to attach. Even if you wash, the antibody-antigen complex will still be there after washing. Then after that, you add 2 drops of anti-human globulin, AHG. This AHG is IgG. The IgG is specific for the antibodies. So it's going to attach to the antibodies. So it's an antibody that can attach to another antibody, but it doesn't attach to the antigen or the red blood cells. So if initially there was a reaction between antigen and antibodies forming this complex, then when you add 2 drops of AHG, it's going to attach to these antigens to the extent that when you centrifuge and then you observe, if these antibodies were still there, and then you can appreciate agglutination, it means that the blood is not compatible. But if these antibodies were not specific for these antigens, after washing, they'll be washed out. Even if you add 2 drops of AHG, it's not going to find any antibodies to attach to. So you find that there will be no agglutination after centrifuging. So you find that there will be no agglutination here. It means that the blood is compatible. So this is cross-matching in the lab. So we proceed now by looking at the rhesus blood grouping system. So the D antigen or the rhesus factor was first demonstrated in rhesus monkey, hence the name rhesus blood grouping system or the rhesus factor. So you can see that it was first demonstrated in the rhesus monkey. It doesn't necessarily mean that the rhesus antigen came from a monkey or a rhesus monkey, but it was just first demonstrated in a monkey, then later on it was discovered in human beings. So the rhesus factor is a red blood cell surface antigen, just like the A and B antigens. The rhesus factor antigen or the D antigen is also a blood cell surface antigen. So it's found within the red blood cells on the surface membrane of the red blood cells. It's an antigen, just like the other ones that we've discussed. So it was first demonstrated in rhesus monkey. Blood groups are classified as rhesus positive or rhesus negative. So it's either you're rhesus positive if you have the antigen for rhesus factor, or rhesus negative if you don't have the antigen for the rhesus factor. So the rhesus factor gets its name from the experiment conducted in 1937 by a scientist, Carl and Steiner, the same scientist, and another one, Alexander S. Wiener. So these are the guys that described the rhesus factor in 1937. The rhesus antigen are encoded by the 2 highly homologous, very similar genes situated very close together on the short arm of the chromosome 9. So these rhesus antigens, or the gene that code for this rhesus antigen, is found on chromosome number 9, just like the ABO genes. They are also found on chromosome 9. But there are different types of these genes, and they are close to each other. So in terms of inheriting them, they are also inherited together because they are very close on this chromosome 9. So the fact that they are so close together means that they are inherited together. One of these, the rhesus D gene, produces the D antigen and its many variants. So the rhesus D gene can code for D antigen and other antigen that are similar to the D antigen. An entire rhesus D gene in D negative people is absent. So in D negative people, the gene for rhesus factor is completely absent. But in contrast, the difference between the CC, EE positive and negative is due to the small changes in an otherwise identical protein. So the other rhesus antigens, like the C antigen and the capital letter C antigen, small letter C and small letter A, these antigens in persons who are negative and positive, they're just minor difference in this protein. Otherwise, it's almost identical. So this has been postulated as a reason for the high immunogenicity of the D antigen. So since for the people who are D negative, they completely don't have this gene. So they don't have it at all. It's either you have it or you don't have it. For that reason, it means that it can stimulate the immune system, because those who are negative, they don't have it at all. So find that they can easily build an immune system against the antigen if it comes into their body. So that's why, in terms of immunogenicity, the D antigen is very important. So there are two antigens that we've discussed, the ABO system antigens and the rhesus antigens. These are the ones that can cause transfusion reaction. Okay, so you can see here that I'm adding another antigen when you're talking of blood typing. So still we're looking at blood typing. So for instance, you want to do a blood typing for this person or for this individual that you don't know the type. So you put the blood. So you have the blood typing plate that has got little wells, and these wells you can put a drop or two of whole blood from a patient or from a person who you want to type the blood. So let's say you put here 2 drops of blood there, there, and there. So on these little wells, you also add anti-sera or antibodies. So here you add anti-A, here you add anti-B, here you add anti-D, here you add a control. The anti-D is for the rhesus positive or rhesus factor. So for this example, you see that here you just have a control where you don't add anything to validate your results. So you can see from here that you have a reaction here, here you don't have a reaction, as well as on the control. So these results are okay, they are valid because you don't have a reaction here just like that. So now when you compare here, it means that your red blood cells, they're reacting with the antibody A or the anti-sera A. So it means you have antigen A. Then your red blood cells, they are also reacting with the anti-D or the antibody D. The antibodies against the D antigen. Then your red blood cells are not reacting with the antibody B or the B antibodies. So you don't have the antigen B on your plasma membrane of the red blood cell. So it means that the blood type for this individual will be blood group A positive. The positive is for the anti-D. So anyone who have antigen D on the plasma membrane of their cells, it's called positive. Like we said, rhesus positive. Those without the antigen, they are called rhesus negative blood type. So this blood type now we are adding a plus. It will be A positive. That positive is the rhesus positive. So using the same, you can get blood from different people and then you put them on the blood typing plate that has got little wells and where you're going to put your drops of blood. So let's say you get blood from 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 people, and then you put your samples here. So you put a drop of blood here, here, and here. Then you put your control. Then even for this person, you forget about the blood types for now. So let's say you don't have the blood types, you want to type this blood. So you put the blood samples like that. For this person, you also put, even for that person, all the way down here. And then on this column, you add 2 drops of anti-A. On this column, you add 2 drops of anti-B. On this column, you add 2 drops of anti-D. Then here, you don't add anything. You use this one as the control. So what would be the blood type for this person here? So you can see for this person, after adding your anti-sera, you are only having agglutination on anti-D, meaning that the red blood cells of this person only has got antigen D. It doesn't have antigen B or antigen A. So it will be blood group O positive. For this other person, there is no reaction on all the antigens, on all the antibodies. So because there is no antigen-antibody reaction, it means that this person is blood group O, but on D, there's also no reaction. So it will be O negative. Then for this person here, you're having a reaction here and here. So you're having a reaction on anti-sera A and anti-sera D. So because of that, you have A antigens on the plasma membrane of the cells. Then you also have D antigen. So it will be blood group A positive for this person. Then for this other person, you can see here you're having a reaction on anti-A. So you have A antigen. So it's blood group A. Because these other ones, they are not reacting. So it will be A negative. For this person here, you're having a reaction here and here. You're having a reaction at anti-B, so it's blood group B, and then on D again, so it will be blood group B positive. So I'll jump to this one here. This one, you're having a reaction on A, B, and D. So because you're having a reaction on all the anti-sera, so it means that this person will have all the antigens. They'll have antigen A, antigen B, and antigen D. So it will be blood group AB positive. For this person here, you're having a reaction on A and B, but not on D, meaning that you have blood group AB negative. Then this person, you're having reaction all the way, including the control. So it means that these results are invalid because even the control is reacting. You didn't add anything to the control, but you have a reaction. So you discard these results or you redo your test because these results are not valid. So that's how you do blood typing. So blood type compatibility table. So it's the same one like the one that I was showing you, but here now I've added the D antigen, whether it's positive or negative. So you have the donor's blood and the recipient. So you can see the donor's blood, O negative, can donate blood to any kind of blood type. It could be blood type O negative, O positive, A negative, A positive, B negative, B positive, AB negative, AB positive. Blood group O can donate that. The reasons of which I've already explained to you guys. So that's why blood group O negative is the universal donor. Then blood group A positive will be the universal recipient because it can receive blood from all blood types. So you can see that. So you can read the summary. This is just the same information. Reasons incompatibility. So the reasons incompatibility, there is a mechanism that you need to understand with the reasons incompatibility, especially for pregnant women. So these reasons incompatibility, you have already seen that if you have antibody against the rhesus antigen, there could be agglutination reaction because the antibodies against the rhesus antigen can react to the antigen. So you have antibody antigen reaction. So this can also take place within the fetus that is developing. So when you're about to get married, you also need to do blood typing so that you know to say that whether you are compatible with your husband or what, because the offspring that you can, or the children that you be giving birth to as a woman later on, you need to pay attention to the rhesus factor because it can bring about rhesus incompatibility. I'll explain how that comes in. So you can see the fetus here, the umbilical cord, and the placenta. This is the uterus. So within the uterus, there is no mixing of the fetal circulation and the maternal circulation because at the placenta, there's gaseous exchange taking place quite all right, but the blood from the fetus doesn't move to the maternal circulation or the blood from the maternal circulation do not move to the fetus. So there is no exchange of red blood cells. But there could be exchange of certain antibodies that I'll explain, especially the antibody for the rhesus antigen. For the antibody for the rhesus antigen, you have IgG. The IgM, like I said, they are big enough so they can't cross the placenta to move from the maternal circulation to the fetal circulation. But the rhesus antibodies, they are smaller enough because they are IgG, so you find that they can cross from the placenta to the fetus. And then that can result into a reaction that will explain. So let's look at the mechanism of rhesus incompatibility. So rhesus incompatibility can occur by two main mechanisms. The most common type occurs when an rhesus negative pregnant mother is exposed to rhesus positive fetal blood. So when the mother is rhesus negative, they don't have the D antigen or the red blood cells, meaning that in plasma, they're going to have anti-D antibodies. So they're going to have antibodies against the rhesus antigen. So if the rhesus negative mother is carrying a rhesus positive fetus, and then there's a mix-up of blood, the mother can start building immunity against the rhesus positive blood. That can result into the baby undergoing a reaction, a rhesus incompatibility reaction. So you can see in this diagram, you have the mother or the female who's pregnant, and then we have a baby developing in the uterus. You have the placenta and the umbilicus here. So if this child is rhesus positive, of course, it's getting that gene from the father because the mother is rhesus negative. So it doesn't pass that gene to the offspring. So this offspring is getting that gene or is inheriting that gene from the father. So if the fetus is rhesus positive and the mother is rhesus negative, like I said, there's no mix-up of blood. So you find that there's no reaction. But for some reason, if there's a mix-up of blood, if this fetal blood is gaining access into the maternal blood, you find that the mother will start building antibodies, or the mother will be immunized or sensitized, and then she'll start producing antibodies against the red blood cells. So those antibodies are small enough to cross the placenta, so they will cross the placenta, then they will move to the fetus, and then they will attack the fetal blood. That will result into hemolysis. So if this blood from the fetus is getting mixed up with maternal circulation, and then the immune cells of the mother will be immunized, and then they'll start producing antibodies against the red blood cells with a rhesus antigen. Then these IgG, they are small enough, they can cross the placenta to go and attack the developing fetus. That will result into rhesus incompatibility. So you have the placenta here, you have the fetal placenta circulation, maternal circulation. So if the fetal blood with the D antigen, so you can see this red blood cell with a D antigen on the plasma membrane, they are crossing to move to the maternal circulation. In maternal circulation, you have maternal antibodies that are produced against the fetal red blood cells. So they will attack these red blood cells. So these antibodies, they are produced in higher quantities. So some of them, they cause the placenta to go in the attack now the fetal blood. That will result into hemolysis of the fetal blood. And then it will result into hemolytic anemia of the newborn. Okay, then the child is born with such condition. But what you need to know is that this reaction takes place mainly in the second pregnancy. The first pregnancy, you won't see any effect. Why? It's because by the time the female she's giving birth, that's where now there will be mix up of blood during the process of giving birth or during labor. So you find that the first child will be okay. Why? It's because you find that she will give birth, and then that's when she will be immunized against those red blood cells. So the second child now will find the antibodies. So the second child will be the one that will be attacked. So the causes for immunization, so you can see here, you have the rhesus negative mother, the rhesus positive fetus, red blood cells that can cause the placenta to go to the mother, and then they'll cause immunization. So what would be the causes of this immunization? What would be some of the factors? So there could be a spontaneous or induced abortion. So when there is abortion, you find that there will be some blood vessels that can break and then can result into mixing of blood, or trauma. If there's an injury, can also result into mixing of blood, or invasive obstetric procedures, and also normal delivery. There could be some mix-up of blood. When that happens, the fetal circulation may leak into the maternal circulation. Sensitization occurs, leading to maternal antibody production against the foreign rhesus antigen. the maternal rhesus immunoglobulins, which are called IgG antibodies against the D antigen, can freely cross now from the fetal circulation to maternal circulation. Then there will be antigen antibody complexes that will form, and these antigen antibody complexes that will form within the fetus, and then the fetal spleen will get rid of these red blood cells. Once you have these antibodies attaching to the antigen, the red blood cells or the fetus can't move within the capillaries of the spleen, and then that will result into damage to the plasma membrane of these cells, and then you have more of hemolysis taking place, and then that can result into fetal alloimmune-induced hemolytic anemia. Anemia within this fetus. Why? It's because the red blood cells are being destroyed before their lifespan. The other mechanism for rhesus incompatibility, so you have the rhesus incompatibility can also occur when a rhesus negative female receives a rhesus positive blood transfusion. So when you're doing blood transfusion, then you give rhesus positive blood to a rhesus negative woman, you'll find that that woman will start now being immunized. She will develop antibodies against the rhesus antigen, and then when she's pregnant, those IgG immunoglobulins can cross and then to go and attack the baby. So this is the other mechanism that can result into more production of rhesus antibodies. So in part, this is the reason that the blood banks prefer using blood type O negative or blood type O rhesus negative as the universal donor type in emergency situations when there is no time for type or cross-matching of the blood. So if you don't have much time to do blood typing or cross-matching, you find that the normal blood that you can use for blood transfusion is O negative, because the O negative is the universal donor, meaning that it won't stimulate the immune cells to produce any antigen against it. So you don't want it to produce antibodies against the rhesus antigen. You don't want it to produce antibodies against the A antigen or the B antigen. So this is the perfect blood to use in emergency, because you don't have to do all those blood typing and cross-matching that will waste time. So what will be the effects of the rhesus incompatibility on the fetus? So what will happen to the fetus? Hemolytic disease of the newborn, also known as hemolytic disease of the fetus or newborn, or erythroblastosis fetalis, is a condition in which the normal lifespan of the fetus's red blood cells is shortened by the action of specific red blood cell IgG antibody, which crosses the placenta from the mother. So it will result into erythroblastosis fetalis. Why is it called erythroblastosis fetalis? It's because when the red blood cells of the fetus are being attacked by the antibodies against the D antigen, most of these red blood cells will be destroyed. So you find that the bone marrow will be very active. So it will start now releasing the red blood cells, the immature red blood cells with the nucleus, which are called erythroblasts. So these erythroblasts will increase in circulation. That's why it's also referred to as erythroblastosis fetalis. So among these antibodies are some which attack the red blood cell in the fetal circulation. The red cells are broken down and the fetus can develop reticulocytosis and anemia. Anemia in the sense that there's a lot of hemolysis, then reticulocytosis because there will be a lot of erythroblasts and reticulocytes in circulation of this fetus. That's why it's called erythroblastosis fetalis. This fetal disease ranges from mild to very severe in fetal death from heart failure, which is also called hydrops fetalis, that can occur. So there will be heart failure. Why? It's because you have hemolysis of red blood cells, and hemolysis of red blood cells will result into production of toxins that can attack other cells. And on top of that, can also block some blood vessels, and then that can also result into heart failure because of less blood in circulation. So when the disease is moderate or severe, many erythroblasts are present in the fetal blood, and so these forms of disease can be called erythroblastoma fetalis, like I've already explained, because you have a lot of these reticulocytes and erythroblasts in circulation. So this disease is also called erythroblastoma fetalis. So you can see here you have fetal hemolysis because of red blood cells being attacked by the antigen, the antibody against the rhesus factor. Then you can also have hydrops fetalis. Then you can have jaundice. Okay, so why do you have jaundice in these neonates after birth? So like I said, the rhesus incompatibility, what will be affected will be the second pregnancy. Why? It's because the first pregnancy